In this session, we will study one of the basic tissue of our body, that is epithelial tissue.
Epithelium is an avascular tissue composed of cells that cover exterior body surface, lines internal closed cavities, including vascular system, body tubes. It also constitutes glands and receptors for special senses. So, epithelium is an avascular tissue. It receives nutrition through the process of diffusion. Then the cells, whenever they cover the exterior body surfaces, we use the term cover. It forms covering over the exterior surface, and whenever it lines the internal closed cavity, we use the word lining for internal cavities. For example, in blood vessels, there are three layers. Innermost towards the lumen is tunica intima, then there is tunica media, and then tunica adventitia. So the tunica intima is lined by endothelial lining. Then the body tubes like hollow viscera or the ducts of exocrine gland, they are also lined by epithelium. Then it constitutes glands. So there are exocrinal and endocrinal glands. When the glands get detached from surface epithelium and form a cluster or clump in the mesenchyma or mesoderm, it is known as endocrinal gland. And when the gland is connected with the help of duct with the exterior or surface epithelium, it is known as exocrinal gland. So these glands are also derived from the epithelium. Then the receptors for special senses. So we know the special senses are vision, hearing, taste, smell. So in case of olfactory area present in the upper part of the nose, the olfaction is again by the pseudostratified epithelium. There the receptors are present. Then in case of tongue, the taste buds are there. So they have taste reception or gustatory receptors. So these are also epithelium. So they form receptors for special senses. Epithelium is derived from all the three germ layers. So we know the three germ layers are ectoderm, mesoderm and endoderm. So from the ectoderm, the epithelium that develops is of oral cavity, nasal mucosa, cornea, epidermis, that is a part of skin, then mammary gland. These are all epithelium derived from ectoderm. Then in case of liver, pancreas and lining epithelium of respiratory and gastrointestinal tract is developed from endoderm. Then the uniferous tubules of the kidney or nephron, then the linings of male and female reproductive system, endothelial lining of circulatory system, mesothelium of body cavity, these are all mesodermal in origin. So mesothelium means the serous lining over the peritoneum, pleura, pericardium, it is mesodermal. Then the endothelial lining of blood vessels, it is also mesodermal in origin. The epithelium has some general features in them. So first is epithelium is an avascular structure and it receives nutrition by diffusion. Then the epithelium is closely opposed and they are adherent to one another by means of cell to cell adhesion molecules that form specialized junctions. Then they exhibit functional and morphological polarity. The As you can see in the figure, the upper part or the apical domain has free surface, and over that free surface some surface modifications can be seen like cilia, microvilli, stereocilia. So cilia are motile projections from the cell surface, and Microvilli are finger-like projections meant for absorption. Stereocilia are again hair-like projections from the cell surface. So the apical surface or the free surface may show some surface modifications. Then the lateral domain is the one which is attached to adjacent cell through junctional complex. So there are variety of junctional complex between the two cells. So mainly there is anchoring junction, then gap junction or tight junction depending on the function. And the lower part or the basal domain or basal lamina is resting on the basement membrane. This basement membrane is again made of two layers. So the upper facing towards the basal part of a cell is known as lamina lucida and the lower part is lamina reticularis. So these cells are resting on the basement membrane and they are again forming a junctional complex known as hemidesmosomes. In previous figure, we have seen that the epithelial cells have apical domain which is a free surface. But there are certain cells, they are closely opposed to one another and that's why they lack apical free surface. So their apical domain is not free but they are closely opposed to one another. And such cells aggregate and it is typically seen in endocrinal glands such as interstitial cells of Leydig in the testes, then luteal cells of the ovary, islet cells of Langerhans and pancreas, then the parenchyma of adrenal gland and anterior lobe of the pituitary. So in these cells we can see the cords or clumps and they do not have the free apical domain. They form epithelio-reticular. Also, it is seen in the epithelio-reticular cells of the thymus. So, it is all depicted in the figure. Then, the organized collection of epithelioid macrophages merge and they form giant multinucleated cells, and such tumors are known as granuloma. Epithelial cells perform various functions based on their location. For example, skin. The epidermis part of the skin protects the body from abrasion, injury, or external environment. Then it acts as a barrier. It permits selective substance to cross it. For again, we'll take example of epidermis. It prevents entry of microorganisms like viruses, but it allows entry of some topical ointments which are lipid soluble. Then it helps in absorption. We'll take example of intestinal epithelium. So the nutrients are absorbed from the digested food with the help of these epithelial lining. Epithelial cells also perform function of secretion or excretion. So in the first diagram you can see that the apical domain has got secretory granules and they have certain secretions which are excreted out of a cell. For example, in case of exocrinal gland like salivary gland, here the saliva is excreted by the lining epithelium. Then in the figure given below you can see two sensory receptors. So first is of olfactory receptors and second is of gustatory receptors or the taste buds. So here the epithelium performs function of receiving sensory signals from external environment. So thus it performs the function of sensory receptors. There are two common ways of classifying the epithelium. First is based on the shape of cells facing towards the free surface of epithelium. So by seeing the shape we can classify the epithelial cells as squamous cells. Squamous means flat cells, then cuboidal cells and columnar cells. Second method of classifying is depending on the layers of epithelial cells. So when single layer of epithelium is present, it is known as simple epithelium, and when more than one layer of epithelial cells is present, it is known as stratified epithelium. Thus we have classified the epithelium by two ways. First is depending on the shape. So when the cells are broad or flat, it is known as squamous epithelium. When the length and width is equal, it is cuboidal, and when the length is more than the width, it is known as columnar epithelium. And the second way is depending on the layers of epithelial cells. When only one layer of epithelial cells is present, it is known as simple epithelium, and when two or more layers of cells are present, it is known as stratified epithelium. In this chart we can see classification of epithelium based on layers of cells. So when a single layer of epithelial cells line a structure, it is known as simple epithelium. So here only a single layer of epithelial cell is there, and it is further divided into simple squamous, simple cuboidal and simple columnar. So these are all a single layer of cell, and the next variety is stratified epithelium. So multiple layer, two or more than two layers of epithelium are present in stratified epithelium. They are further classified as keratinized stratified squamous epithelium, non-keratinized stratified squamous epithelium, then stratified cuboidal and stratified columnar epithelium. Apart from simple and stratified epithelium, there are two modified epithelial cells. So these modification or modified epithelium are pseudostratified columnar epithelium and transitional epithelium. Thus, depending on the layers of epithelial cell, we have first divided the epithelium into simple, having single layer of cell, and stratified, which has multiple layers. Further, we have divided simple into simple squamous, simple cuboidal and simple columnar, and we have divided stratified epithelium into keratinized or dry stratified squamous epithelium. Second is non-keratinized or wet stratified squamous epithelium. Third variety is stratified cuboidal, and fourth is stratified columnar. And also two modified epithelium are seen, that is pseudostratified columnar epithelium and transitional epithelium. In simple epithelium, a single layer of cells rest on the basement membrane, and the simple epithelium is further classified into three groups based on the shape of cells. So first is simple squamous epithelium. It is composed of flat cells. Then simple cuboidal epithelium is composed of cells which have equal height and width. Third variety is simple columnar. These are also known as tall cells. They are composed of cells with height more than the width. In this slide, we can see simple squamous epithelium. So this is the simple squamous epithelium. Here a single layer of flat cells or broad cells are present, and the cytoplasm is also thin layered. So the cell membrane and its cytoplasm stains pink with the eosin, whereas the nucleus is elongated, it is flat, and it produces bulging on the cell surface, and the nuclei is stained dark violet in color because it stains with hematoxylin. So this entire lining is the simple squamous epithelium. It is the endothelial lining in which the cells are broad and flat, and the cytoplasm is forming a thin rim. Both are pink in color, and the nucleus is flat, elongated. It produces a bulging over the cell surface, and the nucleus stains violet in color because it stains with the hematoxylin. Simple squamous epithelium is located in the endothelium. So this is endothelium. Endothelium is the lining epithelium of blood vessel. May it be artery, vein or capillary. So this flat broad cell with elongated nucleus which forms a bulging over the cell surface is the endothelial lining of the capillary. Then the simple squamous epithelium is also present in endocardium, that is innermost layer of the heart. It is also located in mesothelium. Mesothelium is the lining epithelium over serous membrane like pleura, pericardium, peritoneum. Then the simple squamous epithelium is also lining the lung alveoli. So in this slide we can see these are all lung alveoli, and these are lined by flat simple squamous epithelial cell. Then in this slide we can see this is the Bowman's capsule, and this is the parietal layer of the Bowman's capsule. So the parietal layer of Bowman's capsule is also lined by simple squamous epithelium. As the cells in simple squamous epithelium are very thin, they allow easy diffusion across the epithelium. Thus, their important function is rapid transportation of substances, then diffusion of gases, osmosis, and filtration of fluid. It has got applied significance also. In case of anemia with hypoproteinemia, the plasma proteins concentration becomes low, so reverse osmosis takes place and the fluid from venous capillaries will move and it will accumulate into the extracellular spaces and it causes pitting edema. Second variety of simple epithelium is simple cuboidal epithelium. So in simple cuboidal epithelium there is a single layer of cuboidal cells. In the cuboidal cells the height and the width of the cell is almost equal. So when the height and the width of the cell is equal, it is known as cuboidal epithelium. Here the nucleus is round and it is centrally placed, and the cytoplasm is eosinophilic. Simple cuboidal epithelium. It is located in thyroid follicles, small ducts of exocrinal glands, germinal epithelium covering the ovary, choroid plexus that forms CSF in ventricles of brain, inner surface of lens, pigment cells of retina, ducts of Bartholin gland, and distal convoluted tubule. So this slide is of ovary. So we can see the germinal layer of the ovary. This germinal layer is formed by cuboidal epithelium. Then the distal convoluted tubule of the kidney. These are also lined by the cuboidal epithelium. Then this slide is of thyroid gland. So these are the thyroid follicles. When the functioning of thyroid is moderate, then the lining epithelium of these thyroid follicles is cuboidal. Then this slide is of retina. So the first layer of retina is pigment layer. This is also cuboidal epithelium. Main function of cuboidal epithelium is absorption and excretion, then synthesis and secretion of substances. The third variety of simple epithelium is simple columnar epithelium. So in simple columnar epithelium, the height of the cell is more than the width of the cell. So these are also known as tall columnar cells. Here cytoplasm is abundant and it is involved in synthesis and storage of metabolites. Then if we see the nucleus is oval or elongated and it is more towards the basal region of the cell. Also in columnar epithelium towards the apical part we can see some cell projections like columnar epithelium with cilia. It is mainly seen in respiratory tract and fallopian tube. So cilia are mainly motile structures. Then this slide has got the tall columnar epithelium with stereocilia. Stereocilia are static, they are immotile, and these are modified microvilli meant for absorption. And columnar epithelium with cilia are seen in internal ear and epididymis. Then in this figure we can see columnar epithelium with striated borders. So these striated borders are formed by microvilli. Microvilli are finger-like projections from the apical surface of the columnar epithelium. When these microvilli are uniform in size and shape, they form striated border. Striated border of the microvilli is seen mainly in small intestine. In this figure, we can see columnar epithelium with brush borders. So here the microvilli are of irregular size, as we can see some are larger and some are smaller. So they form brush border which is mainly seen in gallbladder. Then this is the goblet cell. So it is called goblet cell because its shape is like the goblet. Here the apical part is extended and the basal part is constricted. The nucleus is oval in shape, and towards the apical part we can see the secretory granules which have mucin. So the goblet cells mainly secrete mucus secretions. Simple columnar epithelium is located in the lining epithelium of the stomach, then intestine and gallbladder. Simple columnar epithelium is also lining the uterus. Then simple ciliated columnar epithelium is seen in fallopian tube, central canal of spinal cord, ventricles of the brain, then some part of respiratory tract, efferent ductules of a testis, then a part of middle ear, auditory tube and the ventricles of a brain. So this diagram is of uterus, and here you can see the lining epithelium of uterus is simple columnar. So the uterus is lined by simple columnar epithelium. And this second slide is of fallopian tube. So we can see the tall columnar epithelium, and in the apical part there are cilia. So it is simple ciliated columnar epithelium which is seen in fallopian tube. Functions of simple columnar epithelium are: first is secretory. So in case of exocrinal glands, it secretes enzymes. In case of goblet cell and mucous salivary gland, it secretes mucus secretions. Then if we see the lining epithelium of small intestine, it is mainly absorptive in function. Then if we take the example of respiratory tract, there we have pseudostratified ciliated epithelium. So these cilia will help in propulsion of mucus towards the exterior. In case of fallopian tube, there is propulsion of ova towards the ampulla of the fallopian tube. Then in small intestine, the microvilli will increase surface area, it will increase absorptive surface of the cell. So the microvilli can be striated as in small intestine, or brush border is seen in gallbladder.
So far, we have seen two types of epithelium depending on the number of layers. So first is simple epithelium, in which single layer of epithelial cells are resting on the basement membrane, and in stratified or multiple layer there are more than two rows of epithelial cells. So a different variety or modified variety which we can see in this diagram is pseudostratified epithelium. Pseudo means false and stratified means multiplied. So there is a false appearance of stratification. So there are two reasons why the single layer of cell is there, still it appears stratified. So as you can see in the diagram, some cells are tall columnar and some cells are small. So the smaller cells are basal cells. So there is difference in the height of the cells, though all are resting on the basement membrane, but some are taller and some are smaller. And the second thing is that in case of basal epithelium, which are small in size, the nucleus of such cells is lying towards the base, and for the tall columnar epithelium the nucleus is lying above. So there is appearance of two or more rows of nuclei, and due to these two reasons, though there is a single layer of epithelium and all the cells are resting on the basement membrane, still there is appearance of false stratification. So the first reason for it is difference in the height of the cells, some are taller and some are smaller, and second is there is appearance of two or more rows of nuclei. So this gives appearance of false stratification, and such type of epithelium is known as pseudostratified epithelium. Pseudostratified ciliated columnar epithelium is located in respiratory tract, mainly trachea and bronchi. So in the first figure we can see there are some tall columnar epithelial cells over which cilia is present. Then there are small basal cells. Then we can also see goblet cells. So its nucleus is also towards the base. Then some serous cells can be seen in which towards the apical part secretory granules are there. Brush cells, they have microvilli over its apical part. Then we can see Clara cells, and from the diffuse neuroendocrinal cell there are argyrophilic cells as they stain with the silver. Okay, so here the height of the cells is different. So in the first figure we have seen pseudostratified ciliated columnar epithelium which is present in respiratory tract. Then the second variety is pseudostratified columnar with stereocilia. So though the name is cilia, in the stereocilia it is not a cilia, it is non-motile and it is modification of microvilli. Stereo means static, so it is immobile, and pseudostratified columnar epithelium with stereocilia is seen mainly in epididymis and vas deferens, and its function is to absorb the secretions from seminiferous tubule. Then we can see pseudostratified columnar epithelium with no cell surface projections in auditory tube and membranous urethra. So in the second figure which is of olfactory epithelium. Here we can see there are tall columnar cells, and then there are sustentacular cells. They show microvilli. Then the yellow colored are olfactory receptor cells. We can also see smaller cells, they are basal cells, and the gland present here is Bowman's gland. So olfactory epithelium is also pseudostratified epithelium. And the third slide is of epididymis. So here there is pseudostratified columnar epithelium with stereocilia. So these stereocilia are modified microvilli, and stereocilia are also seen in the internal ear. When more than two layers of epithelial cells are present, it is known as stratified epithelium or multi-layered epithelium. In stratified epithelium, only the cells present in the basal layer will rest on the basement membrane. And again, depending on the shape of a cells, we further classify stratified epithelium into stratified squamous, stratified cuboidal, stratified columnar epithelium. First variety of stratified epithelium is stratified squamous epithelium. Here several layers of cells are present, and the basal cells will only rest on the basement membrane. The basal cells are also known as germinal cells. So these cells are mitotically active, and they will continuously divide mitotically, so that the superficial dead cells are replaced by the cells that are coming from the basal layer. So the in the basal layer the cells will mitotically divide, and they will migrate to the superficial layer, and the nucleus of these cells are round to oval. And as we go from basal layer to the superficial layer, the size of the cells will go on decreasing, so that in the most superficial layer we get the squamous epithelium, in which the nucleus is elongated and flat. We further subdivide stratified squamous epithelium into keratinized and non-keratinized epithelium. Keratinized stratified squamous epithelium is also known as dry type of epithelium, and non-keratinized is wet type of stratified squamous epithelium. In case of stratified squamous epithelium, the superficial cells show keratohyalin granules in their cytoplasm, and this type of epithelium is dry, and the most superficial cells are dead. They do not have nuclei in them, and such dead cells form a layer known as stratum corneum. So in the stratum corneum, the cells are dead and they do not have nuclei in them, and it is located in the epidermis of the skin. Its function is to protect deeper structures from mechanical injuries. Also, it acts as a barrier against infection, and it prevents dehydration. Another variety of stratified squamous epithelium is non-keratinized. So in non-keratinized epithelium, there is no stratum corneum, and all the cells in the superficial layer are alive and nucleated. And as we go from basal layer to superficial layer, the size of the cells will go on decreasing gradually, and in the superficial most layer we can see it has squamous epithelium. So mostly the basal layer is columnar, then there are 3 to 4 layers of polygonal cells, and superficial cells are squamous epithelium. So the epithelium remains almost always moist. So this type of epithelium is moist or wet epithelium. Non-keratinized stratified squamous epithelium is located in the epithelial lining of oral cavity like oropharynx, laryngopharynx. It is also lining the esophagus, vagina, tonsil, vocal folds, and cornea. So mainly the oral cavity and the structures within the oral cavity like tongue is there, then tonsils. These are all lined by stratified squamous epithelium. Also, the cornea has got stratified squamous non-keratinized epithelium. So in these two diagrams of esophagus and cornea, we can see the stratified squamous epithelium, and mostly it is moist, and the function of such epithelium is to protect underlying structures. It acts as a barrier against pathogenic organism, and if there is deficiency of vitamin A, then the non-keratinized epithelium may start keratinizing. So excessive keratinization may be there due to deficiency of vitamin A. Parakeratinized epithelium is similar to keratinized epithelium. The only exception is that in parakeratinized epithelium, the cell nuclei are present in stratum corneum. We have seen in the keratinized epithelium, the superficial layer is dead. It is known as stratum corneum. It is devoid of nuclei. But in parakeratinized epithelium, the stratum corneum has got nuclei in them. And the example of parakeratinized epithelium is masticatory mucosa located on hard palate and gingiva, or we say gums. Second variety of stratified epithelium is stratified cuboidal epithelium. It consists of two or more layers of epithelial cells, and the most superficial layer of cells is cuboidal in shape. It is mostly located in the ducts of exocrinal gland, some ducts of the sweat gland, salivary gland, and the ducts of sebaceous gland also show stratified cuboidal epithelium. Its main function is that it acts as a barrier, and also it provides passage for transportation of secretions via ducts. Variety of stratified epithelium is stratified columnar epithelium. It is mostly two cell layer thick epithelium, and the superficial cells are columnar in shape. The basal cells are mostly cuboidal and they rest on basement membrane. And such type of epithelium is seen in the lining of some ducts of exocrinal glands like salivary glands, then fornix of conjunctiva and cavernous urethra. Its function is to provide passage for secretions, and it also acts as a barrier. Thus, the stratified epithelium is divided into stratified squamous epithelium. Stratified squamous is further divided into keratinized and non-keratinized. Keratinized has multiple layers. Basal layer are mostly cuboidal or columnar. Superficial are the squamous cells. And here in keratinized epithelium, stratum corneum is non-nucleated and it has superficial dead cell layer. Example is epidermis of a skin. In case of non-keratinized epithelium, it is moist and it has multiple layer. Basal layer is cuboidal, superficial layer is squamous epithelium, and here the superficial layer is not dead, it has nuclei in them, and it is again having the superficial layer is again having squamous cell. Example is oral cavity, esophagus, vagina, anal canal, vocal folds and cornea. The second variety of stratified epithelium is cuboidal, stratified cuboidal epithelium. So it is mostly two cell layer thick. Superficial cells are cuboidal. It mostly lines the ducts of sweat gland and salivary gland. Then stratified columnar epithelium. Again it is two cell layer thick and the superficial cells are columnar. Example is duct of some exocrinal glands, fornix of the conjunctiva and cavernous part of the urethra. Transitional epithelium is a modified epithelium. It mainly lines the urinary tract. So it is also known as urothelium. So transitional epithelium lines major part of urinary passage. And if we see the appearance of the epithelium, it can stretch. So it has extra reserve of cells. So when it is in relaxed form, there are 5 to 6 layers of the epithelium, and when it is stretched it is 2 to 3 cell layer thick. Go layer by layer, we can see the basal cells. the basalmost cells, they are resting on the basement membrane. And above that, we get 2 to 3 layers of polygonal cells. And the most superficial layer has got cells which are of umbrella shape. So when the urinary bladder is in relaxed state, we have 5 to 6 layers of epithelial cells, and the basal cell is cuboidal, then 2 to 3 layers of polygonal, and most superficially we have umbrella shaped cells. They are more eosinophilic due to presence of plaque. If we see the outer surface of such cells, they are covered by intramembranous glycoprotein, and these will make the cells impermeable to the toxic urine present in the lumen, and the surface cells show mitotic activity. So at times we can see more than one nuclei in single cell. Epithelium or transitional epithelium is located in renal pelvis, major and minor calyces of a kidney, ureter, urinary bladder and part of urethra, and its main function is having ability to distend the urinary bladder, and it is because it has reservoir of cells in it. And then it acts as a barrier because of presence of occluding junctions and intramembranous glycocalyx plaques. Goblet cells are simple columnar epithelial cells which secrete mucus, so they have glandular quality. Goblet means narrow base and expanded apical part. That's why they are known as goblet cells. They are goblet like in shape. And if we see with routine HNE staining, the mucus is washed off by xylene. So the upper expanded portion appears empty because the mucus is washed off. The cytoplasm is filled with membrane-bound secretory mucinogen droplets, and they disperse in the cytoplasm. That's why the cytoplasm appears to the periphery. It is shifted by the droplets, and the nucleus is also shifted towards the base. And due to the secretory function, they have prominent Golgi apparatus, and they have more of rough endoplasmic reticulum towards the base. Goblet cells are located in the epithelium of gastrointestinal tract, mainly the large intestine, and also they are present in the respiratory tract. And its function is to secrete mucus and protect the lining epithelium. Stain the goblet cells with periodic acid skeeve, it imparts magenta color to it. And if we stain it with mucic carmine staining, it gives deep red color to the goblet cells. And applied importance of goblet cell is that if these cells are damaged, then it's defective. It makes the mucosal barrier defective, and there is abnormal commensal of bacterial and other infections. So there will be defective host immunity, and the adaptive immune response will also be defective in the respiratory tract. So there will be repeated respiratory infection and intestinal inflammation. Amine precursor uptake decarboxylation is a group of diffuse neuroendocrinal cells. Such cells take a precursor process, and then by decarboxylation it forms amines or peptides, and they serve as neurotransmitters or neuromodulators. And such APUD cells or amine precursor uptake decarboxylation cells. These are oval membrane bound granules with dense core, and such cells are present in the adenohypophysis or anterior lobe of the pituitary gland. Then the neurons of hypothalamus that synthesize oxytocin and vasopressin, and then it is transferred to the neurohypophysis. So they also have APUD cell. Then the chief cells of the parathyroid gland which produce parathyroid hormone are APUD. Then the parafollicular cells or C cells of the thyroid, they produce calcitonin. Adrenal medulla has got chromaffin cells in them. They secrete adrenaline and noradrenaline. Then the cells of gastropancreatico-endocrinal system, like beta cells of islet, cells of Langerhans are there in the pancreas. Then we have gastrin, motilin. So these are all examples of APUD cells. These cells are also located in the glomus cells of carotid bodies. They produce dopamine and noradrenaline. Then the melanocytes of the skin produce promelanin. Some cells of pineal gland, placenta, modified myocytes of the heart also have APUD cells in them. Renin producing cells of the kidney and also the APUD cells, they are producing paracrine and endocrine hormones. Now, what is paracrine effect? When the cells have ability to affect their surrounding cells through their secretions, it is known as paracrine effect. And when we stain such cells with silver impregnation or silver salts, they appear black in color. So they are also known as argentefin or argyrophilic cells. Applied importance of such cells is APOD tumors occur mainly in gastrointestinal tract and lungs. In the lungs, we have Kulczycki cells. They lie close to the basement membrane of the bronchi and bronchioles. Also, the enterochromaffin cells in the gastrointestinal tract and cholecystokinin in proximal part of the small intestine. Their tumor is common. Some of the epithelial cells are modified and they have sensory innervation in them. They are known as neuroepithelial cells. So, in special sense organs, like in the first figure, we can see olfactory epithelial cells. So, here it is pseudostratified, and the yellow color cells are the receptors for of the olfactory area. And the second is of taste buds present in the papillae of the tongue. So the taste bud also has receptors for taste sensation, and they are innervated by sensory nerve fibers. And the third diagram is of macula of utricle and saccules in the internal ear. Here also receptors are present. So these are the neuroepithelial cells present in the special sense organs. The epithelial cells show presence of actin and myosin contractile proteins in them. They are known as myoepitheliocytes, and mostly such cells are situated close to secretory element, and they are stellate shaped or they form a basket around the acini, and they are seen in salivary glands, mammary glands, sweat gland. So when such myoepithelial cells surround the acini of, for example, salivary gland, and when the myoepithelial cells contract, they will squeeze the secretion present in the gland, and the secretion is thus uh, excreted out of the gland by the contraction of such myoepithelial cells. And these are ectodermal in origin, and they contain a protein called desmin, which is specifically present in the muscles. So they are also known as myoepithelial cells. They are innervated by autonomic nerves, and they are located in relation to the acini of glands, then intercalated ducts, intra and extralobular ducts. They are located between the basal lamina and the acini or ductal cells of the serous mucous acini, and they are also surrounding the sweat glands. When one type of epithelium is transformed to other type of epithelium, it is known as metaplasia. For example, normally the esophagus is lined by stratified squamous epithelium, but when such epithelium is transferred to columnar epithelium, it is known as Barrett's esophagus. Mostly metaplasia is a precancerous condition. Then, fine needle aspiration cytology is a technique in which Needle is inserted into the cell mass. After being stained, they are examined under microscope for investigation and diagnosis purpose. Mostly we do FNSC for cell mass like lymph nodes and thyroid. Then there is pap smear. So here the superficial cells of the cervix are scrapped and again after staining they are studied for pathology.